Good afternoon, everyone. Today I'm going to be talking a bit about restoring salt marsh habitat through paired sediment and ribbed mussel addition. So to start off with a little bit of background, as we're all likely familiar with, rapid sea level rise is an increasingly prominent problem in salt marshes. As sea level rise begins to outpace marshes' ability to accrete or gain elevation, flooding also begins to exceed the tolerance of native marsh vegetation. When this happens, there are several distinctive shifts in marsh hydrology. Ponds at the marsh platform start expanding into large pools or megapools, and marsh habitat begins retreating inland, meeting up with coastal infrastructure. This suggests that there's a need for restoration techniques that can maintain marsh elevation at an optimal range for native vegetation. One technique that approaches this issue is sediment addition, also known as thin layer placement or sediment augmentation. In this technique, dredged or quarried sediments are placed on top of deteriorating areas of marsh platform. While this technique has been used in larger areas of marsh in the Gulf Coast and other US regions, there have only been relatively limited small-scale projects using this technique in New England. However, the pilot projects that have been conducted in New England, such as the one pictured in the photos to the right, have shown promising results, particularly in terms of marsh vegetation recovery. As you can see, the little playpen of sediment has a lot of marsh grass after 2 years following the addition. So there is a need for more data that documents marsh response to sediment addition, particularly looking at other factors, including faunal response and the microbial community. One other area for future opportunity in sediment addition is incorporating positive species interactions, such as mutualistic relationships between the Atlantic ribbed mussel and marsh vegetation. Mussels filter feed the water passing through them and contribute to nitrogen cycling in a way that promotes vegetation growth. And they also stabilize sediments through their production of thistle threads, which would be a very beneficial quality in sediment addition, as these added sediments could use additional stability. So with these considerations, I have two primary research questions for my dissertation work. First, how does salt marsh habitat respond to sediment addition, both paired and unpaired with ribbed mussel placement? And my objective here is to document the response of vegetation, fauna, and the soil microbial community to restoration. Secondly, how does marsh response to a small-scale sediment addition pilot project compare with response to a larger-scale sediment deposition? And my objective here is to provide data for coastal regulators in New Hampshire and New England on a larger scale to inform future management work. So my study site is at Adams Point, and at each of the points indicated on the map, I have a block of experimental treatments. And this diagram depicts my experimental design. So at each of these blocks, I have a control plot that's both framed and unframed. I also have a mussel-only treatment, a sediment-only treatment, and then a paired sediment and mussel treatment. Each of these arrays of treatments is associated with a healthy patch of reference marsh. And in the plots that are surrounding the large pool next to the causeway at Adams Point, I have plots both within the mudflat in the pool itself and in the surrounding low marsh. To give a brief overview of the restoration timeline for this work, over the past summer in 2023, I established the experimental design at Adams Point. In May, I got all the necessary permits to do this work, and then in early June, collected pre-restoration data. In late June and early July, we added sediment at a depth of 14 centimeters, consistent with prior work, gathering sediments from a local quarry and adding them manually to the plots, as you can see in that top left photo. In August of 2023, the mussel addition took place. We collected mussels from the marsh edge and tagged and measured them and placed 10 into each mussel treatment, adding mesh for predator exclusion. As I mentioned, I'm going to be looking at vegetation recovery over time in addition to community composition for fauna, specifically benthic macroinvertebrates and the soil microbial community. I'm also going to be looking at hydrology, pore water chemistry, and documenting mussel growth and survivorship. I want to share these data with the regulatory community and public to inform future restoration efforts. And finally, the next steps of my project are going to be doing this on a larger scale at a sediment addition project that's proposed in Wells, Maine. So we have a 2-acre area designated for restoration in Drake's Island, which is shown in the photo there. And this is going to be done in collaboration with the Rachel Carson Reserve and Ducks Unlimited. And I am eager to explore comparing the smaller scale pilot project with this larger scale restoration. And thank you to everyone who has helped with this project. I'm happy to answer questions later.